So our today's lecture is going to be about DNA replication. In our last lecture we studied about the basics, what was central dogma, about DNA, about RNA and the basics. And the first step for central dogma is DNA replication. So many students actually run or try to avoid these chapters because they think that these chapters are really annoying or really difficult or too conceptual. Well, the reason is because they try to cram or try to memorize the theory simply without understanding these topics. So it's really important that you understand these topics, and the best way to understand, to get the concepts, is by visualization. So in this lecture we will try to visualize this concept from as many diagrams as we can. We will look at this process from as many angles as we can. We will discuss about semi-conservative model, about the whole steps for replication, about all the enzymes that are involved in DNA replication, and we will not leave any topic that is related to IMAT. So the most high yield concepts that you have to know for your IMAT exams are in front of you. You can take your screenshot, and then we will move to our first topic, and the first topic is about semi-conservative model. So first of all, let's discuss about what is replication. So the process of self-synthesis of DNA molecules, or you can say making replica of DNA molecules, is called as DNA replication. So this process actually happens during the S phase of cell cycle, right? So you will study about S phase, G1 phase, G2 phase during your cell cycle chapter, but you have to know that this process actually happens during the S phase of cell cycle. So first of all, the molecule of DNA which is replicated is called as the parental DNA, that is right in front of you, and the molecules that are formed after replication, they are called as daughter DNA molecules. So from one parent DNA molecule you will have two daughter DNA molecules. So how does actually DNA can actually replicate, right? So while scientists were trying to find the answer for this question, after so many discoveries, they came to a conclusion that there are three different models to explain the replication process. So the three processes are conservative model, semi-conservative model and the dispersive model. So these are the three models that we will discuss in our today's first topic. So first is about the conservative model. So what actually happens in conservative model is that this model states that after replication has occurred, one daughter molecule will contain both parental DNA strands. As you can see here, after the parental DNA were actually replicated, you had molecules like this. This is in blue color and this is in yellow color. So after the second replication, the result would be that the daughter molecules would contain both parental DNA strands. The other daughter would contain DNA strands of newly synthesized material. As you can see here, the parent was blue color, and the DNA molecules would be actually conserved. As you can see, these were the parents, and from parents the most of it was conserved. You had 3 yellow DNA molecules and 1 blue molecule. So you can see that most of the parental type of DNA were actually conserved, right? If we move on to our next model, that is semi-conservative model. So according to this model, the two parental DNA molecules separate out, and each of these strands then serve as a template. As you can see here, these are the templates here. These are both of the templates, and after these templates, the result is going to be two DNA double helices, both of which consist of one parental and one new strand. From this first template you will get just like the old one and a new one. As you can see here, this is just like the parent and this is actually really new, while this one is definitely like the parent and this is actually new. So as you can see here, the parents or the templates were half conserved while half were completely different. As you can see, the parents were blue and yellow, and one of them was blue and yellow and the other was in yellow color. I am saying these in color formations because students can memorize by memorizing or retaining the data in the form of colors better. If you see colorful diagrams, colorful pencils and colorful writings, then memorizing these kind of concepts would be really easy for you. So in the semi-conservative model, half of the parental DNA is conserved while half of it is like completely new, right? If we talk about dispersive model, that is the last model, that is the most like different from these. So what actually happened in dispersive model is that during DNA replication, the DNA molecules will actually break down into small nucleotides just like this, right? The DNA molecules will break down into small nucleotides, and after that the DNA molecules will be dispersed. Then these nucleotides will combine together again and you will have new nucleotide to form. As you can see here, they will disperse and they will form a new nucleotide again, and from them you will have still really different type of DNA molecules that are completely different from their parents. So this is actually a dispersive model. So when these three models were studied by famous scientists, that were Meselson and Stahl experiment, they proved that the replication is actually followed by the semi-conservative model. So conservative and dispersive models are actually the models that are not true. So the true model is actually the semi-conservative model, in which half of the parental DNA is conserved while half of it is not conserved, and you will get completely new DNA molecules. So it is conserved in semi portion, right? That's why it is called as semi-conservative model. So semi-conservative model is the model that you have to memorize, and this is the model that we will discuss in detail in our lecture. So you can take a look at this table and you can take a screenshot of it. This table would be really important for you. After you have discussed it, let's start our lecture. So if we discuss about the mechanism of DNA replication, so first of all you have to know that DNA replication is actually a continuous process. It does not stop at any specific point. It is always continuous, but for you to understand, for the sake of convenience of your understanding, we will break it down into three phases. The first phase is initiation phase, the second would be elongation, and the third would be termination. We will discuss all of these phases one by one and try to understand the replication process. Again, the replication process does not stop at a specific point, it is a continuous process. So the first phase is going to be the initiation phase. So initiation phase, there is a formation of a bubble, and that bubble is called as replication bubble. As you can see in front of you, there is a bubble like structure, and it is called as replication bubble, and it is formed at a particular site, and the site is actually called as origin of replication. So the site that it starts, this origin, and this is called as origin of replication. So if we talk about the origin, actually the origin is really important, and it is different for eukaryotes and for prokaryotes. If we talk about eukaryotes first, then eukaryotes, you can say, contains more than one origins. So eukaryotes contains more than one origin of replication. But if we talk about prokaryotes, so prokaryotes only contains one origin of replications. So eukaryotes will contains multiple origin of replications, and from these origins you will form multiple bubbles, or you can say replication bubbles. So if eukaryotes have multiple origin of replications, then it will have multiple replication bubbles. If prokaryotes have only one origin of replication, then it will have only one replication bubble. Simple, right? So first of all, let's discuss about how replication bubble is actually formed. So you have to know that there are two types of enzyme. The first enzyme is called as DNA Helicase, and the other enzyme is called as DNA Gyrase. So these are the two enzymes that you have to be really careful about. These are really important. So first of all, if we talk about DNA Helicase, so it actually opens the DNA helix structure. It opens DNA helix. So how does it actually works? If you can see here, this is a DNA molecule in front of you, and if you move both of these strands from each other, like this, you will have a bubble-like structure. So DNA helicase is the enzyme that will open up this DNA helix structure. So the two strands of the DNA can be separated. If we talk about DNA gyrase, so DNA gyrase actually works before the DNA helicase. So how does DNA gyrase actually works? DNA gyrase will actually support, or you can say facilitate, the unwinding of DNA duplicase. So what will it do? It will facilitate the unwinding of DNA. So it will help to unwind the DNA by helping DNA helicase, right? So DNA helicase and DNA gyrase, both of them work together. DNA gyrase will reduce the tension that will be created during unwinding of DNA. When the tension is reduced, the DNA helicase will easily open up the DNA strands and it will form a replication bubble. So now you can see that there is a replication bubble that is right in front of you, but the replication bubble can also form DNA helicase again. You can say that what if the bubble closes again? So to prevent the bubble from closing again, we have an enzyme that is called as single-strand binding protein. So what does this protein do? It will actually bind both of the strands of DNA and it will prevent the DNA from coming back. So the enzyme name is single-strand binding protein. You can say, we will say it SSB, single-strand binding protein, SSBP. So what does SSBP will do? SSBP will actually prevent the DNA to pair up again, right? So it will prevent DNA pairing again. So it will prevent the DNA to pair up again. With the help of SSBP, we can make the DNA or keep the replication bubble where it is. Okay, so these are the enzymes and proteins that were involved in the initiation process. So that was the first step, that was initiation. The next step is elongation, or you can say extension, or you can say the polymerization process. So during this elongation or polymerization phase, this is the phase in which the formation of daughter strands will actually happen. So there are actually two formation or two strands. The first is called as the leading strand, the other strand is called as the lagging strands. So as you can see here, there are two type of strands. When the DNA actually unzips, you will get a leading strand that is in front of you just like this, and you will also have a lagging strand. So these two strands will be formed during the elongation process. So these two strands are actually synthesized by a DNA polymerase enzyme. So DNA polymerase enzyme is going to be really important in our elongation process. So first of all, we will go back to our elongation process back again. Okay, so for this purpose, we have an enzyme that is called as DNA polymerase, but before DNA polymerase we will talk about another enzyme, and that enzyme is called as the primase enzyme. So first of all, we will talk about primase enzyme. So what does primase enzyme actually do? So first of all, let's discuss about this enzyme. So primase enzyme is actually involved to arrange some RNA nucleotides. First of all, it will arrange RNA nucleotides. So what are these RNA nucleotides doing in our DNA replication? So they will actually form these kind of arrangements, specific arrangements on the template strand of the DNA, that the template that we are using for the DNA, and these really short fragments of RNA nucleotides will act as primers. What we will call them? We will call them primers. So primer is going to be really important in this process. So what is a primer? So primer is actually a site, you can say it is the start site. So this is the site where the activity of DNA polymerase actually happens. So DNA polymerase and primase actually work together. So first of all, primase will form the primer, and from the site the primer is formed, the DNA polymerase enzyme will actually work. As you can see here, our RNA and DNA polymerase will work on this site. So first of all, you have to know what is a primer, right? You have to know primer is actually a arrangement of DNA that is present on the starting site. But you have to know another thing. The other thing is that the primase and DNA helicase actually work together in the form of a complex, and they are called as primosomes. So you have studied about DNA helicase and primase. So both DNA helicase and primase form together, and they will called as primosomes. Let me write it down for you. It is called as primosome. Primosome. Easy, right? Helicase and primase, they will form primosome. Primase plus helicase. Both of these combined together are called as primosome. Easy, right? Easy peasy lemon squeezy. So next thing is about, you have to know about DNA polymerase. So what actually and how DNA polymerase actually works. So there are actually three types of DNA polymerases. We will discuss about them one by one. So the first we will discuss about is going to be DNA polymerase 1. So this is going to be our first enzyme that we will discuss about, and then we will discuss about DNA polymerase 2, and the third is going to be DNA polymerase 3. So these three polymerases will work together during your DNA replication process. So DNA polymerase 1, DNA polymerase 2 and DNA polymerase 3. So let's start with DNA polymerase 3. So DNA polymerase 3 is actually the main enzyme, so that's why we are going to discuss it first. So it is the enzyme that actually synthesizes the two strands. So its function is to synthesize your strands that are leading strand and the lagging strand, right? So it will actually start the template for replication process. So it will synthesize the strands, the leading strand and the lagging strands. The other thing is that it also work and do the proofreading at the end. For example, you are done with the whole process. At the end of the process, DNA polymerase will act as a conductor, or you can say as the chief, and it will also do the proofreading of the whole process again. The next enzyme is DNA polymerase 2. So DNA polymerase is actually helps in the repairing of the DNA. So it is like the carpenter of replication process. It actually helps in your repairing of the DNA, right? And if we talk about DNA polymerase 1, so DNA polymerase 1, it actually catalyzes the replacement of RNA. You know that we had formed RNA nucleotides, right? And these RNA nucleotides were acting as a start site and they were called as primer. So RNA nucleotides were at the start of DNA, but we don't need RNA nucleotides in DNA. So DNA polymerase will actually replace RNA primers. RNA would be replaced by DNA nucleotides, right? So these are the three enzymes that actually work during the replication process. So there is DNA polymerase 3, DNA polymerase 2 and DNA polymerase 1. And during the termination phase, you will see that the replacement of DNA primers would be done by DNA polymerase 1. So these are the three steps that we will discuss in detail. Like we just discussed the basics. You can say we just compressed the whole topic into these three small steps. Now we will go into the detail of this chapter, right? Like we didn't complete the topic, but we just made the introduction of replication. Now we will go to deeper understanding of all these processes. So the replication fork, how is it formed, how region of replication actually starts. So we have discussed about how replication fork is formed and how replication bubble is formed. So replication bubble when forms, as you can see, this is our replication bubble, right? And when the replication ends is called as the replication fork. This is the replication fork. just like the end of your utensils, right? How does a fork looks like? It is the end, right? That's why it is called as replication fork, just like your spoon and fork. So replication fork is the end of replication bubble. So it is the point where the replication bubble will stops. So first of all, let's start with the leading strand. If you see, this is the leading strand here. The leading strand is actually going from 3' end to 5' end. So the process will always start from 3' end. When the DNA polymerase starts the work, it is actually works on the 3' end, and it will start to add nucleotide only at 3' end, and slowly the DNA polymerase will keep adding the nucleotide and they will keep moving towards the replication fork. As you can see, the first DNA polymerase is working on the first template and it is going continuously. There is no break here, and it is moving towards the replication fork, and the DNA strand is called as leading strand. But after it has reached replication fork, the other strand, the other unit is working on the other strand, and that other strand is called as the lagging strand. So this strand, it actually synthesizes an other daughter to polymerize strand up to a specific length. So when the units are interlinked here, as you can see, the strands actually have to jump like this and then jump again because of the DNA polymerase in between. So because of these jumps, you will have discontinuous fragments here. The template is going away from the replication fork. If it is going away from the replication fork, then it will have specific hurdles in its path, because it is not going towards replication fork but away from the replication fork. So it will have to jump from these hurdles, and because of these hurdles you will have specific gaps in the strand, and these gaps are called as Okazaki fragments. Let me make the diagram. As you can see here, if we make the strand straight, and then you will have small fragments in between, and these fragments would be called as Okazaki fragments. Why are they called Okazaki fragments? Well, because it was made by or discovered by a scientist. The scientist was actually Japanese and his name was Okazaki. So from his name, because he discovered them, it is called as Okazaki fragments. So they are actually discontinuous fragments, and there is a gap in between these fragments. And how many nucleotides are left? Well, the numbers are actually different for eukaryotes and prokaryotes. If we talk about eukaryotes and prokaryotes, the numbers are actually different. For eukaryotes, the numbers are actually from 100 to 200 nucleotides gap is between Okazaki fragments. If we talk about prokaryotes, there is like a large number, that is about 1000 to 2000 nucleotides are left, and there is a gap of these nucleotides. So they jump from, eukaryotes will jump from around 200 to 200 nucleotides, and prokaryotes will jump from around 1000 to 2000 nucleotides, and there would be a large gap between Okazaki fragments. So these Okazaki fragments will form a discontinuous strand, and that strand is called as the lagging strand, right? So leading strand is continuous, it will not have any kind of gaps, while the lagging strand that is going away from the replication fork will have specific gaps in them. So this was about how a replication bubble is formed, what is region of replication, you have understood everything, what is replication fork, and how leading strand and lagging strands actually work. I hope you have understood this topic until now. The next thing is like the conclusion of everything we have just discussed until now. You can take a screenshot. What is region of replication, how does DNA helicase enzyme actually works, what is single strand binding proteins, what is Gyrase enzyme, what is Primase enzyme, what is DNA polymerase 3, polymerase 1, and DNA ligase. We didn't discuss about DNA ligase. So DNA ligase is the enzyme that actually, you can say, sticks or bind the Okazaki fragments so they don't fall apart. It actually uses ATP to like combine, or like you can say seal the Okazaki fragments so they don't fall apart because they are not continuous, right? So DNA ligase actually is like a gum to like stick the Okazaki fragments. right? So this was about the first concept. The next is, let's differentiate between leading and lagging strands, because this concept is really important in your IMAT like exam, because this concept has been like exam favorite, you can say, and from this topic there are so many MCQs. We have already discussed that the template for leading strand is from 3' end to 5' end. For lagging strand, the template is from 5' to 3'. For leading strand, it is always continuous and it is always towards the replication fork. While lagging strand is discontinuous, it has Okazaki fragments and it is moving away from the replication fork. If we talk about primers, then leading strand only have one primer, while lagging strands have many primers, because you will need like primer again and again. After one jump you will need primer again, then you will have a fragment, that is Okazaki fragment, and then you will start another primer and another primer. That's why lagging strands have many primers, right? If we talk about fragments, leading strand have no fragments, it is continuous, and yes, lagging strand has many fragments that are called as Okazaki fragments. If we talk about the final step for the leading strand, then the final step is that the primer would be removed and the gap will be filled. How the gap will be filled? We have already discussed that the gap would be filled by DNA polymerase 1, right? The DNA polymerase 1 will replace the RNA primer with a DNA nucleotide, right? And for lagging strand, each Okazaki fragment will have a primer removed, the gap will be filled, and it will be ligated by ligase at the end. So these are the differences between leading and lagging strands. So you have to take a mnemonic here. Leading is for lazy, so it will only have one primer, while lagging contains lots of work, so it will have multiple primers. So this is the thing that you have to keep in mind. So this was about leading and lagging strands. Let's move on to next topic, that is the Okazaki fragments, the lagging strand problem. So because DNA polymerase cannot synthesize in the direct direction of 3 to 5' end in lagging strand template, it will move from 5' to 3' end, right? So it will have different segments, these small fragments, Okazaki fragments. So the process is, first of all, the primase will synthesize a short RNA on the lagging strand. The next would be DNA polymerase will extend the primer towards 5' to 3', that would be away from replication, right? We have discussed these concepts again and again. I am repeating all the concepts again so that you don't mess up any of these concepts, okay? If you are thinking that we are doing really repetitive things, you have to know that we are repeatedly doing these concepts so that you don't mess up in your IMAT exam. Then next step is going to be DNA polymerase 1. DNA polymerase will remove the RNA primer and fill it with a DNA nucleotide, right? And the last step would be DNA ligase will seal the neck, and the Okazaki fragments will be attached by a phosphodiester bond so that they don't get loose. So as you can see here, this is the leading strand and then you have a lagging strand. Next, you can see here how replication is actually working. So this is a parental DNA, this is leading strand, and this is lagging strand. This is how replication fork is formed, and you can see this is continuous and lagging strand is actually discontinuous. But the first thing you have to keep in mind is that the overall direction of replication is actually from 5' to 3' end. You have to also keep in mind, like many students get this wrong. So overall direction of replication is from 5' to 3' end. So this is really important to know, because many students actually memorize the direction of leading strand and lagging strand. So the overall direction of replication is actually from 5' to 3' end. So we have discussed so many diagrams. You will get these notes, so you can like look at these diagrams again and again so that you don't forget about how this process is actually happening. The last step is actually the proofreading or the error correction process. We have already discussed that proofreading actually is done by DNA polymerase 3 enzyme. So who does the proofreading? It is actually done by DNA polymerase 3. Okay, so you have to be really careful about this. So DNA polymerase 3 actually does the proofreading. So how proofreading actually occurs? When you have a newly synthesized DNA that contains nucleotide and if there is a mismatch, as you can see here, this is a mismatch because thymine should be with adenine, right? But there was a mismatch between there, you got thymine and thymine instead of adenine. So DNA polymerase will find the incorrect material, it will remove it, and it will add a correct sequence, that is adenine. So this will be done by DNA polymerase 3 enzyme. So this is how proofreading and error correction is done. So this is the mechanism here. You can take a look at it, take a screenshot, and then we will move to our next topic, that is the difference between prokaryote and eukaryote replication. So the differences are in front of you. So first of all, prokaryotes actually contains one origin of replication, while eukaryotes contains so many multiple origins of replications per chromosomes. While if you talk about prokaryotes, the speed of replication is 1000 bp per seconds, right? While eukaryotes actually have a smaller speed, slower speed, that is 100 bp per second per polymerase, but many run simultaneously. So eukaryotes actually has many polymerase enzymes. So even if prokaryotes is really fast, eukaryotes have actually multiple replication bubbles, multiple replications happening at single time. If we talk about DNA polymerase, so prokaryotes main DNA is DNA polymerase 3 and polymerase 1, while eukaryotes have all of the polymerase enzymes, right? If we talk about chromosome structures, then prokaryotes actually have no histone proteins, they have circular chromosomes, while eukaryotes have linear chromosomes, they have histone proteins. Where does it occur in prokaryotes? It actually happens in cytoplasm, because prokaryotes does not have nucleus, while eukaryotes have nucleus, and it happens inside the nucleus, and it happens during the S phase. If we have already talked about Okazaki fragments, so prokaryotes have 1000 to 2000 nucleotides fragment size, while eukaryotes have 100 to 200 nucleotides fragment size for Okazaki. If we talk about telomeres, so telomeres we will discuss about telomeres in your chromosome chapter in cell cycle, while eukaryotes contains linear chromosome ends, that's why they have telomeres, while prokaryotes actually don't even have telomeres, so that is not applicable to prokaryotes. If we talk about replication, then the replication is linked to the cell cycle. In prokaryotes, the cell cycle is in the form of binary fission, right? So in eukaryotes, it is in the form of mitosis or meiosis. So the replication is during the S phase of the cell cycle, or you can say during the interphase. So this was all about difference between prokaryotes and eukaryotes. You can see the diagrams here. The eukaryotes have a replication bubble like this, while prokaryotes have replication bubble like this. So this is the differences between eukaryotes and prokaryotes. The difference is really clear in front of you now in the diagram and in the table. So take a screenshot of this table, is really important, and you have to memorize this table. Okay, let's move on to the last thing, that is about telomeres and telomerase enzymes. So what are telomeres? Telomeres are actually the ends of chromosome. As you can see here, this is the edge of chromosomes, and these tips of chromosomes are called as telomeres. So unlike eukaryotes have linear chromosomes, and they have like a fundamental problem, and that is the problem is that DNA polymerase cannot copy the end of the lagging strands, right? Like the end of the strand is really difficult to copy, because there is no primer extended until the end, right? The primer might fall off from the end. So to copy these edges, we have a special enzyme that is called telomerase. So telomerase and telomere are the solutions of this problem. So what is a telomere? So telomere is like a repeating DNA sequence, right? And it is actually really repetitive, and it is TTAGGG3 in humans, and it is repeated hundreds to thousands of times, and it actually stops the DNA chromosome at one end. As you can see here, it actually ends the chromosome. For example, you are going continuously, and telomere is the place where the chromosome will actually end here. So telomere actually is the place where the chromosome sequence end. So it will cap the chromosome ends, and these sequence cannot be lost without affecting the gene integrity. So telomerase is an enzyme that extends the telomeres, and it carries its own RNA template, and they actually helps to copy the DNA, and this is the solution. The solution is telomere and telomerase. That's why they are really important in your replication process. So telomerase activity and their in different cells are in front of you. You have to know the telomerase activity only in germ cells and stem cells. Okay, though for somatic and scancel cells you don't need to be really like, they are not really important, but still you have to know what happens in germ cells. So in germ cells the telomerase activity is really high because they maintain telomere length, while in stem cells they divide many times and retain the full chromosome length, while in somatic cells they don't actually really work really well, while in cancer cells the telomerase enzyme is actually completely destroyed, and telomerase does not happen during cancer, right? So cancer is actually, you know, already problematic cells, and they actually also affect the telomerase enzyme activity. If you think this is too much, you can skip this topic that is about telomeres and telomerase, but I added it because I thought that it was necessary to complete the replication process. It is not present in many IMAT books, but we still added it because I thought that it was necessary, right? So if we end the whole process, then this is the whole process in one single page. So you can see here the whole process is in front of you. You can visualize the whole process, how leading strand happens. You move from leading strand towards the replication fork, and for lagging strand you are moving away from the replication fork like this. So whole process is summarized in this single diagram. So summary table is also here. First would be the initiation proteins. The initiation proteins will start the replication bubble, and then DNA helicase will unwind the DNA molecule. Then the SSB proteins will keep the single strand and prevent them to come back again. Then we have gyrase, or you can say topoisomerase enzyme. It will relieve the tension and help in DNA helicase enzyme. You can say topoisomerase is also called as gyrase enzyme that we have discussed. And then you have primase. The primase will start the primer or the active site, and then you will have DNA polymerase 3, DNA polymerase 1, DNA ligase, and then you will have 2 daughter helicase. Now the whole process is completed. These are the enzymes and their functions. Now the next thing is about the comparison table between replication and transcription. Our next topic is going to be about transcription. We will discuss this table in that topic. And if we talk about enzymes, the whole quick reference is in front of you. I have made small mnemonics so that you can memorize the enzymes. That is, for helicase is the enzyme that unzip the code, while primase will lay the track. It will start it will give you the RNA primer, so that you know that this is the place that the replication will actually start. DNA polymerase 3 is the one that will help you to build the whole wall of DNA, while DNA polymerase 1 will remove the RNA primer, and then ligase will be the one that will seal the Okazaki fragments, and then you will have topoisomerase enzyme. So topoisomerase enzymes actually help the helicase enzyme to relieve the tension, to help in the uncoiling, right? And you have SSBP that will stabilize so that both the strands of the DNA don't come back together, and you have telomerase enzyme that will actually help to extend the whole process, right? So these are the enzymes and what are their functions, so that you don't like mess up their function in your final exam. So we have summarized the whole replication process until now. I think we have covered every single thing. If you still have any question, if you didn't understand any question, you can write them down in the comments. I will try to answer your any query, any question that you had. We try to make this topic as easy as possible and as extensive as possible. Like, we discussed every single thing that was about RNA replication, so you don't have to worry about like what is relevant and what is not relevant for your IMAT exam. We discussed every single relevant thing for your IMAT, so you are ready to go with this chapter for your IMAT exam. So that was all from my side. Take care. Bye bye.